finalized the lipid metabolism, and today we have the first lecture in amino acid metabolism, specifically nitrogen metabolism and the urea cycle. Let's take a look at what our learning objectives are. You will need to know what the molecular contributors are towards the amino acid pool. Also, you need to understand the concept of nitrogen balance and how and what conditions are inducing an imbalance in this tightly regulated process. Also, you will need to be able to distinguish between transamination and deamination. There are two different processes. Each one of them occurs in a different location, employs different enzymes and coenzymes, and has different forms of regulation. Also, you will need to outline the sequence of reactions that occur in the urea cycle and also be able to determine the location for each one of them, given the fact that urea cycle is a process that occurs in different cellular compartments. Also, you will need to be able to explain the fate of urea in the body and also the fate of ammonia and what happens if ammonia accumulates at levels that become toxic to the body. Let's start with dietary proteins. In order for dietary proteins to contribute to either energy metabolism or the pool of essential amino acids, the protein must be digested down to the level of free amino acids, which can be then absorbed from the intestine into the bloodstream and used in various metabolic processes. The digestion of dietary proteins begins in the stomach with the help of pepsin, which is an enzyme that is active in the acidic environment. Basically, high levels of hydrochloric acid induce a conformational change in pepsin and expose its catalytic site, which allows for the cleavage of peptide bonds and shortening of a large protein molecule into smaller polypeptides. The digestion continues in the intestine, where pepsin is inactivated by the alkaline environment, it is done by enzymes that are contained in the pancreatic secretions. Such enzymes are endopeptidases, which are cleaving peptide bonds that are located in the interior of a polypeptide chain, or exopeptidases, such as carboxypeptidase A and B, which cleave off free amino acids from the C or N terminus of a polypeptide chain. The process continues inside the enterocytes, where di or tripeptides are further digested to free individual amino acids by di or tripeptidases, and once they are in the form of individual amino acids, they can be transported through the portal vein to the liver, where they can enter the energy metabolism, or different other tissues, which can use them for different reasons. What's to remember is the fact that proteins do not have a dedicated storage form, like carbohydrates have glycogen as a storage form, and lipids have triglycerides as a storage form. They are not getting stored for future uses. They are used up in whatever processes that are currently needed for. Let's see now what contributes to the amino acid pool. We have seen before that dietary proteins are digested down to individual free amino acids, and they contribute a small portion to the amino acid pool, whatever is derived from 70 to 100 grams of proteins that are ingested daily. Another smaller portion comes from de novo synthesis of amino acids, specifically non-essential amino acids, because essential ones cannot be produced in the body and they need to be imported from foods. The larger portion of the pool comes from the natural degradation of proteins in the normal turnover that occurs in the body. So basically, endogenous proteins have a limited lifespan and they're targeted for degradation at certain times. They produce amino acids that are contributing to the amino acid pool. However, from that pool, the amino acids are used towards the replenishment of the same endogenous amino acids. Amino acids are also used as building blocks for the synthesis of certain molecules, such as neurotransmitters, or peptide hormones, or nucleotides, or creatine. And whatever is left of it participates as energy source in various metabolism. Basically, after the deamination of amino acids, the leftover carbon skeleton can be targeted towards different metabolic processes, and depending on the metabolic process that they participate in, they have different designations. Glucogenic amino acids are converted into glucose in the process of gluconeogenesis, and further they can be stored as glycogen. Ketogenic amino acids are converted into acetyl coenzyme A or acetoacetate, and they can be further converted into fatty acids or ketone bodies depending on the needs. Let's now remember the common structure of all amino acids. They have an alpha carbon, which has attached with an amino group and a carboxyl group. In the process of deamination, the amino group gets removed, and the nitrogen in the amino group participates in the total nitrogen pool. The removal of the amino group usually occurs in the form of ammonia, which is a highly toxic product and needs to be detoxified somehow very quickly in order to not produce damage in the body. So the nitrogen can also be incorporated into aspartate, which is an amino acid that together with ammonia participates in the urea cycle, which occurs in the liver, and forms urea, which is the primary nitrogen excretion product in humans. So basically the reason for urea to be produced is to detoxify the ammonium that is produced in the process of amino acid deamination. The carbon skeleton, the leftover carboskeleton, we have talked about it. It can be used in different metabolic processes, either for energy or storage, either in the glucose metabolism or in the lipid metabolism. Let's see what happens to the nitrogen that is produced in the process of amino acid deamination. Nitrogen has no storage form, so everything that is produced in the process of amino acid degradation, either of dietary or endogenous amino acids, needs to be used up, either by in the formation of new amino acids or new molecules, or whatever is left over, it needs to be excreted in the urine, either in the shape of the non-toxic urea, about 85%, or in the form of the toxic version ammonium, which also gets excreted in a small percentage in the urine. So basically the balance is constantly maintained in a dynamic and adaptive process. The balance is achieved in a healthy individual where there's an equilibrium between the synthesis and degradation of proteins. However, there can be a positive balance where the excreted portion of the amino acid is lower than the intake of amino acids, and that happens in situations when synthesis exceeds degradation. Such as in situations of pregnancy, of muscle building, healing, wound healing after a large trauma, or during growth spurts. Also, there can be a negative balance in which the excretion of nitrogen exceeds the intake of nitrogen. Usually that happens in situations where protein degradation exceeds protein synthesis, and that happens usually during protein malnutrition, during fasting, during starvation, or in stress response. We have seen before that the main source of nitrogen is the amino group that gets removed through deamination in the process of amino acid catabolism. The nitrogen that is removed from the amino acid is in the shape of ammonia, which is a very toxic product that needs to be detoxified in the form of urea before it gets excreted into the urine. The removal of the amino group from amino acids occurs in 3 separate processes or steps. The first one represents the transamination, in which amino groups from different amino acids get transferred to glutamate. The second step is the step of oxidative deamination of glutamate, which is basically the physical removal of the amino group from the amino acid. And the third one is the conversion of the ammonium into urea in the process that is called urea cycle. Transamination is the first step in the removal of an amino group from an amino acid. This occurs by transferring the amino group from one amino acid, no matter what that is, to an alpha keto acid in a reversible reaction. In the process, another keto acid and amino acid are produced. Basically, you can think of this reaction as a swap of amino groups between two amino acids. This process occurs in the cytoplasm of all cells and is mediated by transaminases or aminotransferases, which are enzymes that are usually specific for the pair of amino acid and keto acid. They use as cofactor pyridoxal phosphate, which is a derivative of vitamin B6, and this pyridoxal phosphate mediates the formation of an intermediate in this reaction, which is called pyridoxamine. The most common donor acceptor pair are alpha ketoglutarate as the keto acid and glutamate as the amino acid. The two most important amino transferases are alanine aminotransferase, which mediates the transfer of an amino group from alanine to alpha ketoglutarate, and in the process they form pyruvate and glutamate. And the second most important aminotransferase is aspartate aminotransferase, which transfers an amino group from aspartate to alpha ketoglutarate, and in the process it forms oxaloacetate and glutamate. Although at first this transfer of amino group from one keto acid carbon skeleton to another may seem highly unproductive and may be unuseful, you have to keep in mind what the acceptors and the products are in this process. The acceptors are usually alpha ketoglutarate and oxaloacetate. However, their substrates or their products are the ones that are important, which are glutamate and aspartate, which both participate in the next phases of urea formation. We have seen before that aminotransferases are intracellular enzymes that mediate the transfer of an amino group from one amino acid to a keto acid. Given the fact that they are intracellular enzymes, usually elevated plasmatic levels of either one of these transaminases signals cellular damage. They have diagnostic values and they usually signify a problem in a hepatic disease, either severe viral hepatitis or sudden toxic injury, which results in elevated alanine aminotransferase to a lesser extent aspartate aminotransferase. However, when there is muscle damage, such as myocardial infarction or acute skeletal muscle injury from trauma, usually the elevated levels are of those of aspartate aminotransferase and to a lesser extent alanine aminotransferase. The second step in the removal of an amino group from amino acids is the oxidative deamination of glutamate. Glutamate, as you remember, was the product of the previous transamination reaction, and in this case it gets deaminated. The amino group gets removed in the form of ammonia in a reversible reaction in which alpha ketoglutarate is also produced. This reaction occurs in the mitochondria of hepatocytes and to a lesser degree of the kidney. and is mediated by an enzyme called glutamate dehydrogenase, which can use either NAD or NADP as cofactors. This enzyme is regulated by the energy state of the body. When there is a high energy state with high levels of ATP, the enzyme is inhibited. However, in the case of a low energy state with high levels of ADP or AMP, this enzyme is stimulated to release nitrogen from amino acids and free their carbon skeletons to be diverted through to other metabolism for fuel production. The urea cycle is the third step in the removal of the amino group from one amino acid. It is basically the major process of disposing of the nitrogen that is produced in the highly toxic form of ammonia from deamination of glutamate in the previous step. This ammonia needs to be detoxified as urea, which is a soluble and non-toxic product that can be easily excreted in the urine. The urea cycle occurs in the liver. However, it's happening in two different compartments of the hepatocyte. It begins in the mitochondria and it ends in the cytosol. The urea is a product molecule that has one carbon, which is derived from bicarbonate, and two nitrogens. The first one derives from free ammonia and the other one derives from aspartate. Both are acquired during the urea cycle. You can see here the major reactions that occur during the urea cycle. There are 5 different reactions. The first 2 occur in the mitochondrial matrix, and they're going to be marked in red. And the other 3 occur in the cytosol, and they will be marked in green in the next slides. The first reaction in the urea cycle is the synthesis of carbamoyl phosphate. This happens when the ammonia that is produced in the deamination process of glutamate in the previous step interacts with carbon dioxide and 2 molecules of ATP and forms carbamoyl phosphate. This reaction is catalyzed by carbamoyl phosphate synthase, which is a mitochondrial enzyme that represents the rate-limiting enzyme for the entire cycle. It is allosterically activated by N-acetyl glutamate, which is formed in a reaction between glutamate and acetyl coenzyme A, and is in ample supply after a highly proteic meal. The second reaction of the urea cycle is the formation of citrulline from carbamoyl phosphate, which was produced in the previous step, which reacts with ornithine to form citrulline and a molecule of inorganic phosphate. This reaction is catalyzed by ornithine transcarbamoylase, and it still occurs in the mitochondrial matrix. Once it's formed, citrulline is transported through the inner mitochondrial membrane into the cytosol in exchange for a molecule of ornithine by using an antiporter which can pump citrulline out of the mitochondria and ornithine from the cytosol into the mitochondria. The third reaction of the urea cycle, and the first that occurs in the cytosol, is the synthesis of argininosuccinate by condensation between citrulline, which was produced in the previous reaction, and aspartate, which provides the second nitrogen for the formation of urea. This reaction is catalyzed by argininosuccinate synthetase, and it uses one molecule of ATP, which gets degraded down to AMP and pyrophosphate. The fourth reaction of the cycle is the synthesis of arginine, which is formed by cleaving argininosuccinate into arginine and fumarate. Argininosuccinate lyase is the enzyme that is catalyzing this reaction, and the fumarate that is produced as a byproduct can be converted to malate and further to oxaloacetate and transaminated to aspartate, which can enter the urea cycle in the previous step to provide the second nitrogen, or Malate can enter the mitochondria and participate in the TCA cycle. The fifth reaction in the urea cycle is the production of urea. So we have finally achieved our goal, which was to convert ammonia, which is a product of amino acid catabolism and highly toxic, into a less toxic and more soluble product, urea, which can then be excreted in the urine. Let's see what happens in this reaction. Arginase, which is an enzyme expressed only in the liver, is hydrolyzing arginine into ornithine and urea. Ornithine is then transported back into the mitochondria in exchange for citrulline and can re-enter another cycle, urea cycle, with formation of a new molecule of urea. Urea enters the bloodstream and is transported into the kidneys, where it gets filtered and excreted in urine. However, a very small portion of urea is diffusing into the intestines, where it is acted upon by bacterial ureases and converted into ammonia, which will then be excreted through the feces. Now let's take a look at ammonia. Ammonia is a toxic product that is produced by most of the cells in the body, and it can be disposed of in different ways. The major way is by conversion to a non-toxic product such as urea. However, it can also be excreted as ammonia in either feces or urine. There are a few different sources for ammonia production. First and major one is glutamate deamination by glutamate dehydrogenase in a process that we covered at the beginning of the lecture. Another source of ammonia is cleavage of glutamine in the proximal tubules of the kidney. Glutamine is cleaved by glutaminase to glutamate and ammonia that is produced here is not converted into urea and is rather excreted into the urine as ammonia. Another source of ammonia comes from the oxidation of neurotransmitters such as epinephrine and serotonins in the form of monoamines. You have probably heard of monoamines oxidase inhibitors, which are first-class antidepressants. Also, ammonia originates in the catabolism of purines and pyrimidines. And another source of ammonia we have somewhat covered before when we were talking about how urea can diffuse into the intestine and can be acted upon by bacterial proteases to form ammonia, which can be then excreted in the feces. However, there's another source of ammonia coming from dietary proteins, which can also be acted on by bacterial ureases, and they can be digested to produce more ammonia, which again is eliminated in the feces. Let's see what happens with the ammonia that is produced in the extrahepatic tissues and how it reaches the liver to be converted to urea in the urea cycle. We know by now that ammonia is a very toxic product and highly insoluble, so it needs to be solubilized and detoxified before it enters the bloodstream and is transported to the liver. For that, it is combined with ATP and glutamate to form glutamine in a reaction catalyzed by glutamine synthase, and glutamine represents the non-toxic storage and transportation form for ammonia. It is used as major ammonia removal by the majority of extrahepatic tissues, with prevalence by the brain, by the kidney, and the muscles. However, there are situations in which the body is unable to detoxify ammonia, which leads to high levels of blood ammonia in a situation that is called hyperammonemia. This can have two major causes. One, hereditary, in which genetic defects in any of the enzymes that are involved in the urea cycle are unable to convert ammonia to urea. And second one is situations in which the liver is damaged by cirrhosis, either induced by alcohol or as a result of viral hepatitis. The symptoms are quite severe given the fact that the ammonia is highly toxic and it can lead to vomiting, lethargic state. If it's the case of a hereditary defect, it leads to poor intellectual development, coma, and even death if untreated. The treatment, the first line of treatment, is by a low protein diet, which decreases the release of ammonia from amino acids by bacterial ureases. There is also a pharmacological treatment which uses either antibiotics to kill the bacteria that produce ammonia, or to chelate the ammonia that is in circulation. You can see here an overview of the entire process that we have covered today. In the first part, you can see all the molecules that are contributing to the amino acid pool. And in the second part, how the amino groups get removed from the amino acids in the amino acid catabolism. They are getting removed as ammonia, which needs to be neutralized, detoxified as urea in the process called urea cycle, and then excreted into the urine.